Hello, I'm Matthew Williams, Assistant Professor of Biology at West Virginia University Institute of Technology. Today I'll be talking to you about building an effective monitoring collaboration in the Piney Creek watershed of West Virginia. This has been a collaborative effort amongst many individuals, many of whom I'll talk about in more detail as I go through this presentation. So the Piney Creek watershed is located in Raleigh County in southern West Virginia. It drains approximately 136 square miles, has home to approximately 48,000 residents. Most of the residents live in the major city center of Beckley. So Piney Creek itself starts in the southern part of the county and flows northward up past Beckley, the major city center, and flows into the New River Gorge National Park. So you may have heard of the New River Gorge National Park recently on the news because it is America's newest national park. So a lot of the recreation and tourism of this national park relies on the New River itself. Again, Piney Creek flows up here into the New River. Again, a lot of the recreational opportunities in the national park are based on water activities such as rafting, canoeing, kayaking, fishing, and so forth. So knowing what's in Piney Creek and flowing into New River Gorge is a lot of interest to a lot of different people. So one of the things that's known about what's in Piney Creek watershed and in Piney Creek itself is based off of a report in 2008 from the West Virginia Department of Environmental Protection that found that all 10 streams that are major tributaries of Piney Creek itself have actually been considered impaired. So in 90% of their sampling events, they found fecal coliform exceedances, meaning that the number of fecal coliform bacteria exceeded some standard level. So these fecal coliform bacteria are bacteria that have been associated with a gastrointestinal tract. In most cases, we refer to Escherichia coli. So this E. coli bacteria are ones that are associated with the human gastrointestinal tract or associated with human feces and human waste. Now, a lot of these exceedances in our watershed we know are coming from failed sewer systems, failed septic systems, or even cases where individuals are still straight piping direct into the creek. Now, fecal coliform exceedances are not the only concern in our watershed. We also have other impairments such as iron impairments, but really the major impairment that we've had concern and been dealing with is that of the fecal coliform bacteria. In 2012, a watershed-based plan was developed by Piney Creek Watershed Association to try to remediate some of these impairments. Major goals of that were to increase education and outreach activities and implement certain projects. Piney Creek Watershed Association has been very good at implementing these projects and outreach activities to the community, and the community has been very receptive of this. So for example, here we have a lot of numerous WVU Tech students that are helping with repair work on a stormwater wetland that was developed by the Watershed Association. And the entire community at large has come together throughout these outreach activities to try to improve the overall watershed. This can be seen through the numerous different efforts of litter cleanups that take place continually throughout the watershed. Knowing that the community and the students were very involved with the watershed and had a lot of interest in the watershed, we decided to take a citizen science and student-based approach to try to target projects and measure results of our efforts on these projects on that fecal coliform problem. So we knew that we needed to have a regular water monitoring regime to be able to understand what the effects were that we were having on the watershed as we developed new projects. We also needed to be able to target where these actual impairments were coming from. So again, we knew that we suspected this would be very successful based off of the previous successes of the Watershed Association and the community involvement that we had seen in the past. So again, this initially was started by 3 individuals. So you see myself right here. You see the environmental scientist, Luke Stevens from Beckley Sanitary Board. You see the previous executive director of the Piney Creek Watershed Association, Danielle Stewart. There's really the 3 of us got together and started trying to lay the groundwork for this project. So in 2018, we started laying out what this monthly water monitoring project would look like. And we decided that we would have 21 sampling sites throughout the watershed, mostly located right near Beckley, so it would be accessible to community members and students. And we would figure out what parameters we were going to measure also. So fortunately, it wasn't just the three of us trying to figure this out. We got a lot of help from the current executive director, Corey Lilly, the associate director of Piney Creek Watershed Association, Jim Fetters, and lots of other community members that were very helpful to getting this project off the ground. So much so that we actually were able to submit the Piney Creek Watershed Association Water Monitoring Program Quality Assurance Project Plan, or QAP, for this project, and got that approved in 2020. Now, the actual sites chosen for this, as I mentioned, are located near the city of Beckley. This is so they can be accessible to community members and the students. Now, we chose sites that we think have high likelihood of being locations of contributing to those impairments. So again, ones that we know are near potential leaking sewers or straight pipes or so forth. And it's not necessarily in Piney Creek itself. So again, here you see Piney Creek heading northward up this direction. You'll notice a lot of these are actually in the tributaries that the DEP found were exceedances in their 2008 report. So by getting multiple sites along each of these subwatersheds, we can get very good insights into where along the subwatershed those impairments are coming from. So again, we've got Cranberry Creek, Little White Stick Creek, White Stick Creek, Beaver Creek, and then Piney Creek itself. So at each one of these sites, in addition to getting our monthly sampling data, we also wanted just to know what the habitat looked like. So we utilized the West Virginia Department of Environmental Protection Save Our Streams initiative to get habitat scores, so what the habitat looks like for each of these sites. So this includes things like embeddedness, sediment deposition, bank stability, and riparian buffer. You incorporate all those together to get the overall habitat score. We found in our 21 sites, we had 8 that were considered marginal, 9 that are suboptimal, and one site considered as having a poor habitat. So obviously we want to try to improve the habitat scores of these also. A lot of this initial site selection and habitat score was really driven by 2 students, Dane Gazer and Katie Stanley, who were essential to getting this project. With our monthly monitoring, we get several characteristics. Some of the things we get are just characteristics about the waterway itself, recording things like clarity and color and how much algae there is. We also record weather. Then we always get a general flow observation at the minimum. So we always record what the flow looks like generally. Several months we'll actually take flow meters out and get an actual velocity. We'll also even do channel shape. It depends on if we need that for that sample that given month. Again, we've gotten that characterized for multiple of our sites at this point, but we always at a minimum get a general flow observation. We also always get temperature, oxidative reduction potential, pH, dissolved oxygen both in percent and parts per million, conductivity, total dissolved solids. We get all of these measures with our multi-parameter multimeter. So the one we use is from Hanna Instruments, has been very successful for us for the past 2 years. We also get a total suspended solids grab sample that we'll analyze in the laboratory for suspended solids, and importantly we always get that fecal grab sample too. So with our fecal grab sample, we take that back to the laboratory at the university, where we use these coloscan kits to enumerate our fecal coliforms. These coloscan kits are great because in addition to total coliforms, we can get E. coli numbers also, and they're very approachable for students. It gives a colorimetric based readout. The students can just count the number of colonies of certain colors and figure out how many coliforms there are, and also how many E. coli there are for a given sampling site. Now, one of my favorite outcomes from this project is that of the student and volunteer outcomes. So over the past 2 years, we've had over 30 students that have been completely trained on our protocol and most of them assist us for multiple months. Once they're trained on it, they'll come back for multiple months. They can be involved with the field sampling, the lab sampling, or the data management side of things, and many of them end up being involved with all 3 aspects of the project. So the field sampling, they get to learn how to use the field equipment, they do the sampling techniques, data recording. If they're helping with the laboratory, they can help with the calibrations, the fecal coliform methods, whether setting up the plates or counting the plates. Some of them also help with the total suspended solids data. Now, some students really like the data management side of things, so helping with the data records, so just getting the data input and recorded. And some of them are really good with analysis also. And some of them will take that and turn it into research projects or senior theses themselves. Again, many students get involved with all three aspects of this, and then some just only one or two if that's really what they're interested in. What's been fun is it's not just this generation of students that are getting involved. Some of our students have brought their children out. Some of our community members have brought children and grandchildren out also. So that's been a fun part, seeing the next generation getting involved, going out. Again, this is one of my favorite parts of the project, are those student outcomes. So we've had 5 students that have actually gotten jobs after graduation directly related to the work from the project. 4 have actually used their research projects to help get into graduate and professional schools. 3 have actually gotten internships with the project. 7 community members come out regularly and assist our monitoring efforts. And you see the metrics here for our actual number of hours and our dollar value. Interestingly, we actually had an increase in volunteer hours during the 2020 pandemic. Even though we had to shut down sampling for 3 months, we still ended up with more volunteer hours in 2020 than we did the previous year, and we're on track to have 21. Now, I'm not going to get into all the details of the data with this project, with this presentation, because it's more about the overall project than the actual details of the data. We'll save that for another time. But what's been great about the monthly monitoring is the fact that we can actually discover where the sources are of our impairments. So we know we've got these impairments, but where are they actually coming from? So by doing the monthly monitoring, we can see, which sites are giving us impairments. So something like non-7 in 2019 was giving us impairments. Non-9 was giving us 20 impairments in 2019. And we see that kind of shifts for 2020. So we see that non-9 is not really as much of an impairment now, but non-5 is. So we start tracking these impairments and trying to figure out what's going on. And what we're seeing here, again, this red line is just what our cutoff for. Anything above that's considered an exceedance. Anything at that or below that is not an exceedance, is within reasonable values. So we can track not just the annual average, because that's what that's showing here is the annual average value. By doing it monthly, we can see temporal effects also. So for example, one trend we've noticed is that most of our high numbers are coming in this August and September sampling dates. So if we were just doing once per year sampling and we happen to sample here, we're going to get a lot more exceedances than if we sample in, say, March. So we can start to learn things about our watershed itself, like when it is that we're seeing these exceedances, like in that late summer, early fall. And we see that again for 2020. You see some of those same trends, that late summer, early fall seems to give us those exceedances. Again, we did have to shut down sampling for the pandemic, but we got right back. Also, by getting out and doing the monthly sampling, we can actually get eyes on the watershed and see what's happening in the watershed. So for example, here we have a sewer system overflowing into one of our sites. Same thing here, we can actually see toilet paper streaming out of this sewer overflow and down into the creek. And then sometimes you see odd things like a car in the creek. Again, we're not sure exactly what happened here, but we reported this. Again, it was kind of concerned because there were actually footprints here. Not sure if somebody washed into the creek and then got out of the car, or someone was just investigating the car, but whatever the case is, by being out in the watershed every month, we get to see eyes on what's actually happening out in the water. So again, the data analysis part of it is important too. So what are we seeing in 2019 and 2020? So with the 2 years of our sampling, what is nice to see is actually we're seeing a reduction in the number of exceedances. So back in 2008, that report from the West Virginia DEP saw that there were exceedances in 90% of their sampling events. We're actually seeing about 58% of our 58% of our sampling events had exceedances in our 2 years of monitoring. So again, this is nice to see that compared to what it was in 2008, we're actually seeing a reduction in those exceedances. Now, by sampling in the subwatersheds, we can actually see which one of those subwatersheds had greater exceedances than others. And we can do other analyses like compare it to the recreational water quality indices and figure out, do we actually meet those recreational water quality indices? And we actually see that in most of our subwatersheds and most of Piney Creek, we do meet the recreational water quality standards. So that's really good. It's looking good for the watershed. So what is the future of the watershed research? Continued water monitoring on a monthly regimen, but also we're trying to move towards continuous monitoring. So we've gotten some funds to try to implement remote monitoring stations. So if you're interested in learning more about that, I definitely encourage you to check out Jim Fetter's presentation on remote monitoring stations. It's going to be in the continuous conundrum section, and you can see the title of the presentation there. But eventually we are going to publish all this data and share the data online. Thank you very much for listening to my presentation. If you have any questions, feel free to direct them to me.